Now that you've completed your blood coloring sheet, I just want to go over some common tests that are run that have to do with blood. Plasma levels, and remember plasma is that fluid matrix of blood, has things dissolved in it. So we said it's mostly water, it's dissolved electrolytes, and it has all these different types of proteins in it. Usually a clinician will order a metabolic panel. This is going to tell levels of the electrolytes. It's going to tell them, they're going to measure things like BUN, so those nitrogen containing wastes, and they'll be able to assay kidney function. There's also going to be nutrients in the plasma, so this is where they're going to measure blood glucose levels. So whatever's in plasma is going to be measured through a metabolic panel. I took this table from your notes, and this has to do with the cellular component of blood. And the cellular component of blood is going to be measured by doing what's called a complete blood count, a CBC. And then we get really nitty gritty on the white blood cells, we could do a differential count. I'm going to kind of walk you through a CBC with differential lab results. But when we're going through this, think of the function of each of these formed elements. We know that red blood cells, their primary function is to carry oxygen. They also carry our other respiratory gas, carbon dioxide, which is our waste gas, but to a lesser extent. We know that platelets have to do with blood clotting, so hemostasis. And our white blood cells are our immune fighters, so they protect us from disease. I'm hoping this makes sense to you. There's some terms that go with imbalances. And so when we're talking about imbalances, either these formed elements are going to be too high or they're going to be too low. There's some root words or etymology of these terms that stays the same always. Whenever you see emia at the end of anything, it always means in the blood. When things are too high, they kind of use one or two, depending upon which one sounds better, in all honesty. They'll use cytosis, like leukocytosis, or philia, which means to love, like neutrophilia. Both of those, if you see those at the end of any word, it means that levels of whatever's before it are too high. I'm going to get very general because we're just learning here. Because hematopoiesis, just in case you forgot that word, and hematopoiesis is the formation of blood cells, occurs in the bone marrow. If the bone marrow has cancer and is overproducing cells, you could see high cell counts. The other way would be if cell counts are low, and the term for that is penia, and penia means poverty. So I gave you this picture of this guy and hope to help you remember that. Because the blood cells come from the bone marrow, if the bone marrow is damaged, let's say you have cancer patients, so either through chemotherapy or radiation treatment, you're probably going to see some penias, some low blood cell counts with your cancer patients. I do want you to know that there's a type of infection that can cause either elevated levels or depressed levels. All right. So when a CBC is ordered, it means complete blood count. And it'll give counts of the different formed elements. When it says with differential, that has to do with only white blood cells. The differential part is a white blood cell thing, and I'll cover this at the end. So this first page that I'm showing you, we talked about red blood cells. We know there's stuff with hemoglobin, and a hematocrit is a way to measure that packed red blood cell volume. And I told you just for regular purposes, an average of 45% packed red blood cell volume is a normal hematocrit. We'll talk about the other ranges in just a second. All right. So we know that red blood cells carry respiratory gases, particularly oxygen. And for each of these, there's a normal value. So when lab results come in, usually it's going to show for the patient, it's going to show up green if they're in the good range, and then there'll be a little flag if they're either high or low. For all of these, red blood cells, hemoglobin, and hematocrit, if it's on the low side, you're going to see anemias. So A means without. And so people are going to appear pale, and so that's why they called it anemia, because it looks like they lost their blood. If they're on this high range, then it's called poly, which means many. Cyte, which means cell, and emia means in the blood. And you might think, hey, great, more red blood cells means more oxygen carrying capacity. And that's true if it's in the normal range. If it gets too high, in case you're wondering, like, how high is too high? So let's say their hematocrit is 65%. That's pretty high or higher. You see normal range, and this is values for a female, so 46.6. So if it's really high, there's risk associated with that. If there's too many cells in the blood, it gets very viscous. It gets very thick and it doesn't flow, and it increases the risk of inappropriate blood clotting. That can lead to stroke and heart attack. Let's talk about anemia because it's very common. Anemia is more of a condition than a disease. It really just means low oxygen carrying capacity. The human body uses oxygen in cellular respiration to make energy, and we use energy in the form of ATP, so cells can do work. So if somebody is lacking energy, the number one symptom or sign is going to be a fatigue. Because they're lacking oxygen, then they may also have shortness of breath, particularly when they're exerting themselves. The hemoglobin in blood has pigment to it and gives a person their color. So they may have pallor, which is just paleness. And even if someone has a dark complexion, you can sort of see this paleness on their lips and inside of their mouth. And then it's also sometimes accompanied with heart palpitations. There's lots of different causes for anemias. Kind of makes sense. If somebody doesn't have enough red blood cells, they're going to be anemic. If they don't have enough hemoglobin, they're going to be anemic. And then there's also genetic anemias like sickle cell anemia or thalassemias. So when looking at a hematocrit, which is when you take blood in a centrifuge and spin it down, notice that there's different values for males and females. And notice that it is higher for males. A normal hematocrit for males is 47% plus or minus 5%. Okay, there's always just going to be a range for homeostasis. And for women, for females, it's 37 to 47%. So as long as blood is within that range, that's considered normal. If it's below that, we said that's anemia. And if it's above that, then it's polycythemia. The reasons for this difference, and it's not a pathological difference, it just has to do with testosterone actually stimulates red blood cell production, and also men don't menstruate, so women in their reproductive years have blood loss. There is a baseline level of a hormone called erythropoietin, so right here, and it's abbreviated EPO, that is going to maintain a level of red blood cells. If blood oxygen levels get low, then there will be low oxygen in the tissues, which is hypoxia. And it's going to be the kidneys. They have a way to detect low oxygen levels as the blood goes through the kidneys. The kidneys filter blood and make urine. And it would be low because there's low red blood cells, there's low hemoglobin, or there's not enough oxygen. This could occur because the patient has respiratory disease. Maybe they've picked up this cardio routine and they're stressing their muscles more and their muscles are needing more energy, or they've moved to a higher elevation. When there is hypoxia, particularly in the blood going through the kidneys, then they are going to release erythropoietin. The other organ that releases erythropoietin is liver, but like I said, to a smaller extent. Erythropoietin, it's a protein hormone. Its target is going to be the hematopoietic stem cells in the red bone marrow, and they're going to respond by making more red blood cells. Those red blood cells are going to be released into the blood, and they're initially released. They're kind of immature. They're usually released when they're 1 to 2 days out from being completely mature, and those immature red blood cells are called reticulocytes. And in some blood panels, those are also measured. Once there is more red blood cells, you think this is also going to increase oxygen carrying capacity, and then that should lead to homeostasis. So red blood cells are actually not really cells. They don't have a nucleus. They're actually called corpuscles. They are stuffed full of hemoglobin, and so this is a hemoglobin molecule, and it's made up of 4 protein chains. At the center of them is a heme group. So this is what heme looks like. Heme is the pigment that gives blood its color. At the center of heme is iron. So Fe is the chemical symbol for iron. Iron is a cofactor. What it's going to do is it's going to reversibly bind oxygen. It's going to pick up oxygen in the lungs, and when blood circulates to the tissues, it's going to let go of oxygen. That oxygen goes into tissues, and then the tissues make ATP, make energy. The reason carbon monoxide, so one oxygen, is poisonous to us is that it binds to iron and it doesn't let go. And so it stops the ability of hemoglobin to carry oxygen, and that's how people die of carbon monoxide poisoning. So if somebody's red blood cells are low, their hemoglobin is low, and their hematocrit is low, that tells a clinician that person is anemic. The next question is, why are they anemic? And that's where this next series of tests comes in, and that is called red blood cell indices. You don't need to know these, but I'm just going to kind of tell you what these are. So a red blood cell doesn't have a nucleus, so the word for that is a corpuscle. And so that's what these tests measure. So this one is mean corpuscle volume. So how big are the red blood cells? Are they full? This next one is an average amount of hemoglobin in the corpuscles. This one is the mean amount of hemoglobin in the corpuscles. And this one has to do with how similar they are in size. So if it's really high, that means they're of different sizes. So let's look at some causes of anemia real quick. So we know if somebody has low red blood cells, they could just be losing them, right? They could be bleeding. They could have hemolytic anemia. So let's say they have mismatched blood. That just means they're making red blood cells, but they're lysing. Or they could have something called aplastic anemia, and that is when there's a damage to the bone marrow. But that would kind of show up in the low red blood cells. When it comes to dietary factors, it could be iron that they're missing. They also need vitamin B12 to make DNA. You'll often see what's called pernicious anemia in the elderly, and those of you going into dental hygiene, you can kind of see that on their tongue. They get pale oral mucosa, and they sort of have this little strawberry tongue appearance. Or they could have a folic acid deficiency. So dietary factors, the body's going to make red blood cells, but they're not going to look right. And so some of those corpuscle volumes can tip off the clinician that way. And then there's genetic mutations that lead to anemia. So a very common one is sickle cell anemia, and that is a genetic disease where there's a base pair substitution and the cells have a sickle-like appearance. And the sickle-like appearance, they don't carry oxygen as well. They tend to lyse. They can also cause a sickle cell crisis. They tend to get caught up in blood filtering organs. They can cause damage to the spleen. They could see that. And then there's a whole lot of thalassemias. That's a genetic type of anemia. But you can kind of look over here at the red blood cells. You can kind of see they're all different sizes. So that would show up in the red blood cell indices. So that complete blood count says a whole lot about the red blood cells. Number one thing they're looking for is going to be anemia, because that's the most common. And then too high is going to be polycythemia, and that's not good either. So the next form element we're going to talk about are platelets, and platelets are involved in blood clotting. And we have a whole lot of platelets per microliter, and it kind of makes sense, okay? So if someone has low platelet values, that term is thrombo, because that means clot. Cyte means cell, and penia means poverty, meaning low cells that involved in clotting in the blood. So that means a patient will see bruising under the skin. They can have internal bleeding. They're going to have increased clotting time, bleeding gums. Also think in females. So extra heavy menstrual flow. If someone has too many platelets, and now we're on the high side, thrombo means clot, cell, too high. And what we don't want to happen is inappropriate blood clotting, because we know that that can lead to stroke if it's blood supply going to the brain. It could lead to heart attack if it's part of that coronary circulation going to the heart muscle, and deep vein thrombosis. So just you're going to get too many thrombi or clots if platelets are too high. There is a hormone called thrombopoietin. Is the hormone. It's made by the liver. Its target is going to be the red bone marrow, because that's where blood cells are made. And so that is through negative feedback that is going to control the number of platelets. So we know that white blood cells, that's your immune fighters, so they fight infection. And I'm a rounder, so 5 to 10,000 per microliter. I think that's the number I gave you in your notes. If someone doesn't have many white blood cells, so that's called leukopenia, and you can think that these people are going to be immunocompromised. What that means is they're going to get sick, okay, because they're lacking immune fighters. If it's high, then it's called leukocytosis, so high white blood cells. This is a normal response to infection. If you are under attack, you need more soldiers, so white blood cells go up. If they're super high, like off the chart high, like 150,000 white blood cells per microliter, then that's leukemia, and leukemia is a white blood cell cancer. We colored our way through the different types of white blood cells, and we learned that each of them has a special job. So a complete blood count, if then they ask for, or the clinician orders with differential, then the readout is going to basically give the proportions or the amounts of the different white blood cells. And I gave you mnemonic. This is going to be never let monkeys eat bananas. And so highest concentration to lowest concentration as far as white blood cells. Ooh, I did that badly. Neutrophils, if neutrophils are high, neutrophils are seen with an acute bacterial infection. And it may be high with other infections, but we're keeping it general. Neutrophils are very good at fighting bacterial infections. Lymphocytes, they are assassins to viral infected cells, so you often see those go up with a viral infection. Monocytes and neutrophils are big eaters. Monocytes grow up with chronic infections. And eosinophils and basophils, they can both be seen with some different types of autoimmune and allergies, but eosinophils for sure are always going to go up with a parasitic worm infection. And basophils, what I wanted you to remember is that they are involved in inflammation. So they have a lot of histamine, so think allergies and autoimmune diseases. Like I said, the different percentages of these white blood cells is part of a differential white blood cell count. Hope this helped you with the review, and see you soon.